Out of the nearly 8 billion people alive on planet Earth right now, no other person is exactly like you. From your physical traits, to the health conditions you have, to determining your ancestry, and even to the diseases you might one day be susceptible to. At Illumina, scientists and engineers are working on the forefront of an exciting field called genomics. They're developing cutting-edge technology like this DNA sequencing machine that can decode exactly what makes you, you. This information has the potential to improve our lives and the world we live in. From fighting cancer, uncovering rare diseases, better understanding viruses, to growing more sustainable crops, and even helping to identify and save threatened and endangered species. Today, we are going to learn about genomics and how it decodes the language of life. Hey everyone, I'm Vinoir. Welcome to this Illumina and Discovery Education virtual field trip, coming to you from the Illumina campus in San Diego, California. Illumina is a biotech company that creates and builds machines that can rapidly and accurately sequence, or read, DNA from living things. The information that comes from these DNA sequencers has the potential to revolutionize the field of genomics, and as we'll learn today, improve the world on so many levels. But first, what exactly is genomics and DNA sequencing, and why do they hold so much promise for us? Let's get some answers from Jason Button.
Thanks, Vinoir. When you study genomics, you're not just studying humans. You actually can study anything that's living. All living creatures on Earth have a genome, so you can study single cells, or you can study any animal, plant species, any living organism. Genomics is the study of the genome, which the genome is essentially just all the DNA that's found within one organism. DNA stands for deoxyribonucleic acid. A DNA strand contains small base levels called nucleotides, and these bases are often referred to as A's, C's, T's, and G's. So the nucleotides A, C's, T's, and G's stand for adenine, thymine, guanine, and cytosine. So if you think of DNA, you can think of a strand of DNA being a sentence in a book, and the nucleotides, those A's, C's, T's, and G's, being the letters that are making up that sentence. When you're thinking about the genome, it's almost like a whole instruction manual on how to build the body and make you, you. And so it's really important to know that if there's any mistake in those instructions, then that could cause an impact to your body. It's really important for us to study DNA because there's a lot of information that we can gain from it. One, you can gain information about your ancestry, but on an individual level, you can start to look at differences between your genome and others to see what are the causes for certain diseases in humans. How can we make plants grow in harsher environments or resilient to more diseases? We can also start to learn about the differences of organisms, and that might impact how we save endangered species. Here at Illumina, we build sequencers, which are the instruments that actually read the DNA, and then we build the software as well that will help analyze that DNA sequence and help scientists make decisions based on it. We're also involved in the chemistry behind those instruments. We can sequence DNA from any living organism. So when you start out with a new species, you really don't know what letters are supposed to be where. So over the years, we've compiled what are called reference genomes for a species, and this just means that something that we can compare new sequences to check where the differences are. So for humans, we have a human reference genome, and that really started with the Human Genome Project. The Human Genome Project was a project developed to sequence the first human genome. Up until that point, we didn't have a reference genome for humans. This project cost $3 billion and took 13 years to complete. Now, 20 years later, we're looking at runs that can sequence a human genome, 48 of them actually, in about 44 hours, so under 2 days, and it costs less than $1,000. A huge amount of technology and innovation went into making this a reality.
It's amazing how far DNA sequencing has come and how much more accessible it is. But what do we do with all this information? Let's learn more about the human genome from Obi Onachulum.
I became interested in genomics at a pretty young age. My family, unfortunately, had been affected by various health issues. And so healthcare was always something that I was very passionate about. I loved really trying to understand the basics of why people respond the way they do to different diseases and different medicines. And genomics is a huge part of understanding healthcare. Genomics isn't only about healthcare. Genomics can also be about just the study of genetics. Genetics is what is the basic information of what makes us who we are. People refer to the genome as the language of life because the genome contains all of the information that's going to make up a person or any living thing. So the genome is comprised of 6 billion letters. So a genome is made up of DNA base pairs, and so you can think of it as 3.2 billion base pairs or letters. The genome is huge. If you were to sit at a desk and type the entire sequence of one human genome for 8 hours a day at 60 words per minute, you would be typing for about 50 years. Our genomes are 99.9% similar. Now that seems pretty high, but because our genome is comprised of 6 billion letters, that means that over 6 million letters can be different from person to person. So that 0.1% of difference between one person to the next means that if you look at your friend or if you have a sibling, oftentimes our siblings can look completely different. One sibling could have dark brown hair and the other one could have red hair, or one has curly hair or straight hair. One can be very tall, one can be a little bit shorter. All of those things are things that are expressed from our genes. So each human being has about 20,000 genes. And so you can think of it like a gene is going to determine whether or not you're going to have blue eyes, whether or not you're going to have green eyes, whether or not you're left-handed, right-handed, things like that that are all very unique to each person. Genes contain genetic information about each human being. That genetic information is going to be important. Also, there's a lot of information that we can gather to understand about treating different diseases and different illnesses, or why someone might have an allergy and another person doesn't. That is all information that's going to be contained in the genes. The things we knew about the genome even 20 years ago is like a minuscule of what we know now.
It's exciting how much we can learn about ourselves from our genome, and how that tiny 0.1% makes all the difference between you, me, and every other human on Earth. Now let's check in with Darcy Whitlock to learn how genomics is helping us get better at fighting viruses, including the infamous coronavirus.
I think everybody should know about viruses because viruses can have a bigger impact on not only our health, but just our daily lives than really any other organism. I think a lot of viruses are misunderstood. You think about COVID-19, flu, HIV, they're bad. They cause a lot of disease and a lot of heartbreak. But viruses can do so much for people. There's some types of viruses that don't cause disease at all, but they actually can help cure other diseases. Viruses are actually really important for life on Earth because about 50% of the oxygen that's produced on the Earth comes from plankton in the ocean. But the plankton have to compete for resources with bacteria that are also in the ocean. And there are viruses that will kill off those bacteria so that the plankton can then thrive to produce all the oxygen we need on the planet. A virus is a really tiny parasite that will invade a cell, maybe a cell in a human body, and then replicate. And viruses are technically not alive. They meet a lot of the criteria for being alive. You know, they reproduce, they evolve, but they can't do it on their own unless they invade another cell or somebody's body. Most viruses like to stay in their own host species, but some viruses can move from an animal to a human or from a human back to an animal. We call viruses like this zoonotic viruses. And these viruses are particularly dangerous because typically humans have never been exposed to them before, so our bodies don't know how to fight off that infection. COVID-19 is a great example of a zoonotic virus. COVID-19 not only infects humans, we've seen it infect a lot of large cats, like lions and tigers. Also mink and white-tailed deer all carry COVID-19 now. Genomics has played a really big role in the COVID-19 pandemic because it's enabled us to track how COVID-19 is mutating and how it's turning into new variants. Epidemiologists learn a lot by using genomics. Epidemiology is a branch of medicine that studies all the data generated by human illness. It's a very modern field of study, but it also goes back further than you'd think. The first epidemiologists, I would say, are the ancient Greeks, because they really started studying medicine and trying to understand what made people sick. But what we consider modern epidemiology started in Victorian London. There was a huge cholera outbreak there. And back then, people thought that cholera was transmitted in the air with bad smells. They didn't know that it was in the water. And there was an epidemiologist, Dr. John Snow, and he started tracking every single case of cholera and marking it on a map of London. And by doing that, he was able to pinpoint where exactly the outbreak started. And other doctors at the time in Victorian London thought this was a crazy idea. But he showed that by using data and tracking patients and tracking illness, you can tell where a disease is. And that's really the foundation of what modern epidemiologists do. Before COVID-19, there really was no concerted effort around the world to study a virus. Individual labs might study it, a country might study it, but COVID-19 brought together labs around the globe. And we're seeing more advances in the fields of virology and epidemiology because of the COVID-19 pandemic than I think anybody could have hoped for. It's really exciting to think for the future that we have this foundation to build from.
We've spent a lot of time talking about human DNA and how it contributes to our health and overall appearance. But keep in mind that every living thing has DNA. This includes other animals, trees, crops, birds, fish, and even coral. Let's hear how Khalil Lawless and Leslie Matsushige are using genomics to improve research on plants and animals, sustainability, and conservation.
One of the most common ways that people are using our technology at the moment is to optimize their breeding programs. So they're really trying to learn as much about the genomics of the animals that they're breeding or the plants that they're breeding so that they can improve those plants and animals in order to secure our food systems and feed the planet. So we use the same technology to do a study of animal and plant genomics as we would for human genomics. Only the techniques are sometimes a little different. Plants and animals are different from us. Plants often can be significantly more complex than humans. The sequoia tree is an example of an extremely large genome. So the human genome is around 3 billion bases, but one copy of the sequoia genome is 30 gigabases. So we're talking 30 billion individuals, A, T's, C's, and G's. And so the complexity housed within that genome is vastly more than in humans. So if you think we are the most complex, highly evolved thing on the planet, think again. So agricultural genomics is, as it says it is the combination of genomics and agriculture. An example of improving our food supply in that way is making, a wheat crop that is hardier, that can survive a drought or a harsh environment or is resistant to pests. There are lots of things that threaten our food security, and there's a lot of variabilities in the environment. And the more we can do to make our plants and animals that we depend on robust and productive and resilient to those systems, the better off we'll be. We really do need to produce a lot more food. In the next 30 years, we're going to have a growing population, we're going to have a lot more hungry mouths to feed, and we don't have a bigger planet with which to do that. So we have to drive a lot more efficiency from our food systems in order to meet the growing demands of the populace. And if we don't do that, we're going to have real and very serious food shortages in the next few years. After all, we live on this planet and it sustains us, and we need to work out how to better manage it and be good stewards so that we can sustain it. So agriculture is often focused mostly on the specific plants and crops that we grow and the animals that we have husbandry for and produce for us. But those animals and plants don't exist in isolation. They are grown in natural environments. So when it came to understanding biodiversity in our natural environments, ecological surveys have been the norm, and this is really an observational method or sometimes even a destructive sampling method in the ocean. You have to pull up the fish from the bottom of the sea to see what fish are there, and in the process, you kill them. And so we've been really looking for ways that are easier to scale to monitor biodiversity and minimally invasive as well, so that we're not damaging the environment by observing it at the same time. And so eDNA is something called environmental DNA, and this is DNA that's shed into the environment from the organisms that live there, And if we're sampling that environment and we take up a little bit of soil, or if we take up some water from a stream, it gives us, through sequencing, a way of understanding all of the different types of organisms that are present in that environment, as well as the abundance of those organisms. So we're seeing a lot of community science projects starting to leverage genomics, and there are citizen scientists now that are running around sampling the world and sending all of those samples back in. And I'm excited that we'll get a chance to do that even today and go down and sample the ocean. And I can't wait to see the biodiversity that's present here in our ocean systems in California. So genomics can be used in environmental conservation, in biosecurity, in biodiversity management, but it is also increasingly being used to manage and rescue species that are close to extinction. It's very difficult in a breeding program to revive a very small population with a limited amount of genetic diversity. But if we understand all of the genetic diversity that's present in that population and we can make very careful mating choices, we can hopefully revive some of those populations.
Hi everyone, I'm Leslie Matsushige. I'm an associate curator at the Birch Aquarium at Scripps. Genomics helps the sustainability here at the Birch Aquarium by really allowing us to have successful propagation programs. The animals that I oversee at the aquarium that is my primary focus is the sea dragons and the seahorses. Genomics has helped us in our breeding programs, I would say especially with our sea dragons, because there is a relatively small population of sea dragons in captivity. So the gene pool is not that big in captivity. And so if we can find out how closely related individuals are at different aquariums or even within each aquarium, it'll help us to get proper breeding pairs together that then might be more successful breeders and produce stronger offspring that are more able to survive in a challenging environment. I see genomics helping future conservation efforts by using all of the new technology in genomics. For instance, the eDNA. To be able to detect a species in a body of water is just a fascinating thing to me. And it's going to be mainstream. If we can do stuff like that now, I just can't imagine how much more we will be able to do in the future, and that how we will be able to help species survive and thrive.
We've learned some amazing ways genomics can improve our lives. And believe it or not, that even extends to our mental health. Let's talk about our gut, which absorbs nutrients that support our body's functions. Did you know that our gut is sometimes called our second brain? That's because there are trillions of microorganisms or microbes that live in our gut, and they have a direct impact on our mental well-being. Let's hear from Dr. Priyam Singh to learn more.
Illumina helps study the microbiome by sequencing the DNA of the microbiome. A big portion of the microbiome sits in the stomach, but the microbiome is all over the body. You find them on your skin, in your nose, in your mouth, in your stomach, large intestine, small intestine. In fact, it's like your body, and there's an invisible another body of microbes that's living inside you and on top of you that you're not even aware of. So there are a lot of very interesting facts about the microbiome. The first one is that there are about 100 trillion cells of microbiome in your body. Your body itself is composed of only 10 trillion cells, so there's 10 times that many, which is really cool. Second thing is looking at the diversity. There's just so much diversity, even more diversity than an Amazon forest, because there's so many different species of microbes that we carry in our gut. The microbiome has an effect on every aspect of your health. If you have a good, rich, diverse microbiome, you're gonna feel energetic, you're gonna feel happy, you're gonna feel motivated, you're gonna have more dopamine, you're gonna have more serotonin, you're gonna sleep better, you're gonna thrive. There's a big connection between the brain and the gut. So let's talk about this from an intuitive point of view. You might have seen that when you're very stressed, that your stomach is going into knots, or when you're very nervous, you might feel like you can't really eat. And we also throw these phrases around like, there's butterflies in my stomach. We even say things like, my gut instinct is that we should do this, right? So you can imagine the vagus nerve to be a nerve that is sitting on your stomach and it's spreading all the way from the stomach to the brain. And the nerves are spreading all over, all the organs, lungs, everywhere. And this is the gut-brain axis. So there's this constant communication happening all over your body between the microbiome and different organs. And that's why they play such an important role in human health. In fact, 90% of the serotonin that's made in the human body is made in your gut. Serotonin is the hormone that you need to be happy. If you're feeling sad and depressed, it's because you don't have enough serotonin. So you need to go for a run, or you need to do something to uplift your mood. If your microbiome is not in symbiosis or it's not in balance, then it's going to create these hormones that are going to be harmful, or it's going to create less of the hormones that your body needs in order to thrive. When you're stressed, you create these chemicals like cortisol. You have more inflammation and your immunity goes down. And all of that has an effect on your microbiome. So a great tool to help with stress management is breathing. But most of us, we breathe very shallow, right? We don't use our full lung capacity. So the breathing technique we're going to do is where we will use our full lung capacity. We're going to use the stomach, the lungs to breathe fully. And we're going to do something called the square breath. The square breath is where you breathe in for a count of 4, hold for a count of 4, breathe out for a count of 4, and then hold again for a count of 4. I can show you one right now. So we always breathe through the nose. In this breathing technique, we're going to breathe fully with our stomach. So we can put our hands on our stomach and just watch how I breathe. As I breathe in, my stomach expands and even my chest expands. Watch this. And when I breathe out, the stomach contracts. And I'm going to slowly throw a count out, and you can keep your eyes closed and just breathe with me. Breathe in, 2, 3, 4. Hold the breath. 2, 3, 4. Breathe out. 2, 3, 4. Hold the breath. 2, 3, 4. Maybe do it 5 to 10 times. You will see that there's an immediate response in that your mind has settled down and you have basically moved away from the stress that you're feeling into a much calmer zone.
Thanks, Priyam. We've reached our last stop on this virtual field trip, and it's time to consider the future of genomics. What's ahead for this incredible field of science? Diana Tuman offers some possibilities.
Genomics has the ability to really revolutionize the way that we approach medical care, the way that we understand human diseases today, the way that we could drive towards proactive medical management, and the way that we could really address some of the challenges in today's healthcare. Once a person gets their genome sequenced, we don't need to resequence a person's genome. The DNA of a person does not change. It's something that we're born with. What can change is our understanding of the human genome, and our ability to analyze and find novel information from the information that we've obtained is what changes. And that's the exciting thing about genomics, is the richness of data that we collect from around the world is what helps to drive discoveries. Genomics allows individuals to think in advance of any symptoms manifesting and really treating individuals ahead of time. Proactive medicine is really fascinating and revolutionary, which is powered by genomics because it allows us to anticipate symptoms and diseases before they advance and are in that stage where we can actually treat them and have and offer better rate of prognosis, survival, and improving human health. Innovation is critical to the field of genomics because that's where we thrive. It's the patents that are on the walls here and behind me in Illumina is what drives our instrumentation, our understanding to be faster, to drive deeper sequencing, to make availability of our technology to be cheaper, more accessible. Genomics can really help you tailor and think about innovative ways to transform your future and be at the forefront of it to ultimately take control of the available information and be sort of the stewards of your own lives.
What an amazing day this has been. I've learned so much about genomics, and I hope you have too. My head is still spinning about how much bacteria is in my gut. I never knew about the idea of a second brain and its effect on stress, sleep, and my overall mental health. Genomics also holds so much potential for combating hunger, fighting cancer, finding cures for rare diseases, and more. I can't wait to see what the future holds and how it will impact each of our lives. I'd like to say a huge thank you to all the experts who spoke to us today and to Illumina for hosting us on its campus in San Diego, California. On behalf of Discovery Education, until next time, I'm Vinoir.